Hello, my name is Shyh-Dar Li. I'm Associate Professor at UBC Pharmaceutical Sciences. My research focuses on drug delivery technologies. Today, I would like to talk about Cellax technologies we developed a number of years ago, and using this opportunity to introduce this project to prospective graduate students. So this technology is to address a critical clinical challenge with cancer chemotherapy. As we all know that the current cancer chemotherapy induces a lot of side effects, very toxic. And also we are using a lot of chemotherapeutic drugs that are not water soluble. So for those poorly water soluble drugs, we need to include different formulations to improve the solubility. The problem is that normally those formulations are toxic to human body. And so that's why we come up with the solution with Cellax delivery technology. So we are hoping to improve the targeting of chemotherapy to tumor, but also use this technology to improve the solubility. And this technology is using very friendly formulation, as I will introduce in the next slide. So this shows the chemical structure of Cellax and Cellax nanoparticles. So if you have some chemistry background, you probably can appreciate that Cellax is composed of polysaccharide conjugates. So we modify a natural occurring polysaccharide and conjugate the drug to the side chain of the polysaccharide. So we carefully control the chemistry and that the final polymer conjugate can self-assemble into nanoparticles. The key point is that we want to maintain the balance between the water solubility and the lipid solubility of the final compound. So the particle size about Cellax nanoparticles is about 120 nanometer, but by introducing different chemistries and different conjugation ratios, we can actually control the size down to 20 nanometer. So the first data I want to show you is how efficient the delivery is by Cellax. So this is looking at the delivery of taxane to a tumor. So the y-axis is the taxane amount in the tumor, and the x-axis is the time after injection. And we compare three formulations. The first one, red line, is our formulation Cellax. Blue line is Taxotere, is the taxane drug dissolved in a formulation containing detergent and solvent. So it's not very friendly to human body. The green formulation is Abraxane, is also a nanoparticle form of taxane, but is not chemically conjugated. It's a protein-based formulation. So as you can see, our Cellax nanoparticles outperforms the other two formulations by 10 to 20 fold. But how specific our Cellax nanoparticle targeting is shown by this figure. So this is a model of lung containing small spreads of cancer. So the lung tissue contains a lot of small dots of cancer spreads. So in this case, we want to see if our Cellax nanoparticles actually deliver to the small spots of cancer rather than the whole lung tissue. So I'm going to walk you through the images. So if you look at the blue image, DAPI stands the nuclei of the tissue. So you see very intense nuclei staining and a lot of cells. And Ki67 is the green signal, stands the proliferative cells, meaning tumor cells. The normal cells usually they don't proliferate that much, so you see that you don't see a lot of the green signal in the normal tissue. And so in this green channel, you already can differentiate the tumor cells from the normal lung cells. And move to the red channel, and it's dye. It's a fluorescent dye that we put with Cellax nanoparticles. So now we can overlay these 3 different channels and see where our nanoparticles go in this lung tissue section. So you can appreciate that our nanoparticles are highly associated with the tumor tissue rather than the normal lung tissue, again indicating that Cellax is highly selective for tumor. So next, we really want to study if Cellax can inhibit cancer spread, because as I mentioned, cancer spread is the cause of death for cancer patients. And it's very difficult to treat because once cancer is spread, they are everywhere and very, very difficult to deliver your drugs and to treat those spreading cancers, nodules. So in the first case, we study breast cancer spreading to the lung. So this is lung section and looking at the nodule or tumor load in the lung after different treatments. So in the control, you can see this piece of lung actually contains a lot of nodules. So they are stained as more blue or purple compared to the rest. So you see a lot of dots in the tissue section, indicating lots of tumors. And we have next we have docetaxel DTX treated lung. You see there's no improvement. And next is Abraxane. There's some improvement in terms of the cleanness of the lung, indicating reduce of the tumor nodules in the lung. But look at the final group of Cellax. The lung is very clean, indicating Cellax can inhibit cancer spread. In the nano model, we're looking at the tumor spread to the bone, the prostate tumor to be specific. Again, very difficult to treat disease because once prostate cancer spread to the bone, they eat up the bone and causing fracture, pain, a lot of complications. So as you can see in the control group, the bone has been eaten up, and you see fracture there. And in the middle, we have a free drug, and you see the bone is well preserved now, better preserved. But you see shades there, indicating tumor is still growing there, but just in a slower rate. But look at Cellax bone. So the bone is much preserved, and you don't see shades there, indicating that this is a well preserved bone, and there's little evidence of cancer spread. And the ultimate goal for cancer therapy is to prolong patient survival. And in this case, we are really interested in looking at whether Cellax can prolong survival of animals bearing resistant ovarian cancer. Again, this is a very difficult to treat disease because these patients or animals, their ovarian cancer already spread throughout their abdomen. Cancer chemotherapy drugs are very difficult to reach those tumors, and they are very highly resistant to different therapies. So the curve is a survival curve. So in the y-axis is percentage of survival. So we started with 100% animal, but later on you can see the curve just going down, and at the end we don't have animal survival, so that's 0%. So if you see the curve shifting to the right, that means the animal survival is improved. So we compare Cellax with other different standard treatments, including control, the red line, PLD is also a standard treatment. The blue line, PPT is another standard treatment, is the gray line, and our Cellax is black line. And the final one is PTX. So long story short, only our Cellax nanoparticles is able to improve the survival of these animals. And it's improved from 50 days of survival to 70 days. So very significant improvement. Because of the technology is very advanced, so there's a significant interest in commercializing this technology, and the goal is really mature this technology to a stage that it can be used in human to fight against cancer, especially advanced and resistant cancer. So this is a working model of academia and industry partnership. And I would like to introduce this to graduate students just because, we do a lot of translational research, and what we really want to do with these research is use them to help patients. And I hope all my students will appreciate why we are doing this kind of research. Okay, so the model is that we develop the technology in academic setting. So for example, Cellax technology was developed at UBC and OICR, my prior institution, and later on we formed a company called NanoStar, and then we licensed this technology from academia. And then with some incentive, like licensing fee, and then we take over the technology to the company, and the company will do the following commercialization development, such as how to scale up the nanoparticles to enough amount that we can do detailed toxicology study, and maybe also pharmacokinetic study and other type studies to convince that this is a really good technology that has significant potential for the use in human diseases. And again, the final goal is to make the technology and the product mature enough that they can be used in human. Okay, again, I want to thank you for watching this video. I would like to have volunteer students to help me improve these videos. So if you are interested in science and you are familiar with video editing and animation, please contact me. I would like to work with you. And thank you. I will see you next time.